Okay, good morning or afternoon, everyone. It's morning here in Memphis. As Michal said today, I'd like to talk to you all about some of the work I've been doing as a postdoc here at the University of Memphis and also a little bit at Iowa State with my advisor, Jim Edelman. And yeah, we've been looking at the effects of tolerance in house finches and mycoplasma, and I'll talk about that a little bit today. So as I'm sure you all know, emerging infectious diseases arise when novel pathogens successfully transmit through host populations. So prominent examples include avian influenza, avian malaria in Hawaiian honey creepers, white nose fungus in bats, and chytrid fungus in amphibians. And the emergence of these novel pathogens really does pose an ongoing threat to both domestic and wild animals. These pathogens can decrease population sizes, can contribute to local extirpation and extinction of animal species. So we need to really be able to predict and control the threat of these emerging diseases to better understand how animals can adapt to novel pathogens whenever they arise in their systems. And there are really two main ways we think about animals adapting to novel pathogens. So the first way, I think, is the more common way to think about how animals adapt. So this is resistance. And of course, resistance is all about decreasing pathogen load. So a more resistant individual is going to have a lower pathogen load than a less resistant individual. These resistance mechanisms are generally well characterized in animals. They include the innate and adaptive immune system. Like I said, it's all about decreasing pathogen load for resistance. And we generally think that resistance can really evolve quite rapidly in the face of a novel pathogen. So for example, one example that's already been talked about a little bit this week, but in European rabbits, they really quickly evolved resistance to myxoma virus after they were exposed to it. However, animal populations can also adapt to novel pathogens via tolerance. So this is less well understood in animals, and tolerance does not necessarily reduce pathogen numbers, but instead is all about minimizing damage or cost of infection for a given pathogen load. So if we look at the relationship between pathogen load and damage, we can see that a more tolerant individual, which is shown here by this blue dotted line, is going to have less damage or less cost of infection for any given pathogen load. So if we look at a pathogen load, this individual or population will have less cost of infection, less damage, for instance, for that given pathogen load. So tolerance is really important in quite a few systems we're finding out. So recent work across all kinds of animal taxa has demonstrated that tolerance can arise in response to novel pathogens. And this is important because tolerance can really play a role in preventing extinctions of wild populations. It may increase or decrease the spread of pathogens through populations, depending on the specific mechanisms that tolerance takes. So it's really, really important to understand tolerance in order to understand how animals are adapting to novel pathogens and thus how we can control these pathogens. So despite these recent advances, we really still do not understand the underlying mechanisms of tolerance in animal systems. They're generally not well characterized in most systems. And I just want to give you a few examples of how tolerance might arise in different systems, so the different mechanisms of tolerance. So one mechanism might be damage avoidance. So for example, hosts may decrease costly immune responses, such as inflammatory responses, which can cause immunopathology. In addition, animals may have better damage repair. So an individual might be able to, for example, more quickly replace red blood cells if pathogens cause anemia. And this would be an example of tolerance because of damage repair. And there are lots of other ways that animals might be tolerant to infection, changes in behavior, for example. And importantly, many of these mechanisms are not mutually exclusive. So all of these different mechanisms and the fact that they might work together makes tolerance really difficult to characterize in many systems. So my goal was to better understand the potential mechanisms of tolerance. And to do this, I've been looking at tolerance to Mycoplasma gallisepticum, or MG as I'll refer to it here, in house finches. So MG is a recently emerged bacterial pathogen in house finches. And in finches, we see that MG causes really severe conjunctivitis, strong inflammatory responses. And this is sort of your typical house finch after they've been infected. So they go from healthy to having severe conjunctivitis, and then you can see the infection is starting to resolve here. So this is a pretty typical path for house finch once they've been infected with MG. And there's evidence that this conjunctivitis due to MG can decrease predator avoidance behaviors. And it's possible that it also has other costs, such as decreasing foraging, which is something that's still being explored. So house finches are common across much of North America. There's native populations in the West and introduced populations in the East, and also on the Hawaiian Islands. And MG originally spilled over from domestic poultry into an Eastern house finch of populations in the early 1990s. So around 1994, we believe, MG spilled over from domestic poultry into a population in Virginia. And then after that, MG really spread rapidly throughout the Eastern United States. And it was found in most Eastern populations within just a few years. And then more recently, MG has begun to spread to Western populations. So beginning in the Northwestern United States, and then kind of going down the coast there into the Southwest. And there are potentially still pockets of the Southwest where we don't really find much MG at all. And then in the introduced populations on the Hawaiian Islands, MG is not present there. So this is a completely naive group of populations on those islands. So after MG arrives in a region, which is shown here on the x-axis, the percent of expected population size really decreases as much as 50 to 60%. So you can see here after arrival, we see strong population declines. And this suggests that MG has been a really strong selective force on house finch populations where it has arrived. So recently, what we wanted to do was determine whether there are differences in tolerance to MG across house finch populations. And we did this by comparing 7 different populations, denoted here by these stars, of house finches, and just wanted to see how these different populations responded to experimental infection. And as you can see, we chose populations that span this full temporal range of MG endemism. So we have everything from very early infected populations where it's been for more than 25 years, all the way to the populations on the Hawaiian Islands where MG has not arrived yet and hopefully will not arrive. So we captured individuals. These were juvenile individuals naive to MG, and we did experimental infections with birds from each of these populations. And we measured tolerance as the relationship between maximum pathogen load and maximum I-score, which is a score of that conjunctivitis. So how much damage do individuals have during infection? And more tolerant populations will have a shallower slope. So with a higher maximum pathogen load, maximum I-score does not increase so much. Whereas with our less tolerant populations, we expect a much steeper slope. They have a lot more damage for any given pathogen load. So here we see our maximum pathogen load on the x-axis, and then maximum I-score here on the y. So I-scores go from completely healthy to severe conjunctivitis in these birds. And you can see in our eastern populations, as well as in Washington, birds have lower I-scores, so less damaged from infection, less conjunctivitis, than our Western populations where MG has been for less time. So despite having very similar maximum pathogen loads, we really do see this separation in how much damage individuals or these populations have during infection. So if we were to look at a bird from the east, they might look something like this, a bit of mild swelling during infection as compared to a bird from the west, including the Hawaiian Islands, where they have really severe conjunctivitis and get extremely sick. And keep in mind that both of these birds likely have very similar pathogen loads, so they're paying different costs for those same pathogen loads. So we know that there are population differences in tolerance to MG. So we have generally more tolerant populations in the east, where MG has been endemic for longer, and less tolerant populations out west, where it's been for less time. We also know that there are differences in inflammatory signaling between eastern and western populations. So here we see inflammatory signaling as the ratio of cytokines here. So the lower on this y-axis here, the less inflammatory signaling this population has. When individuals are uninfected in our controls, there's no difference between our eastern and western populations. And this was done in 2013. So you can see Alabama had MG for about 15 years at that point, and Arizona had no MG at all at that point. And then whenever these populations were actually infected, we see that Alabama maintains very low inflammatory signaling, whereas Arizona, those western populations that we now know are less tolerant to infection, have much higher inflammatory signaling. So a dampened inflammatory response may be an important mechanism of tolerance in house finches, but really we don't know a whole lot about all of the different potential mechanisms and how tolerance might come about in these finches. So my goal here was really to explore potential mechanisms of tolerance in house finches to MG. So what are these underlying mechanisms? So I used transcriptomic data, RNA-seq data, to try to determine what genes are differentially expressed across house finch populations that differ in tolerance to MG. I predicted that there would likely be many genes associated with tolerance. As I've said, tolerance is a very complicated trait that can involve a lot of different mechanisms. So it's likely polygenic, and a large number of differentially expressed genes are likely to pop up between our control and infected individuals. The other thing I predicted was that immune and in particular inflammatory genes would be much more upregulated in our less tolerant populations. Given the nature of the pathology of this system, as well as previous work that I just talked about, this seemed a likely prediction. So I quantified gene expression in 4 of our populations. Virginia and Iowa were our more tolerant populations, and then Arizona and Hawaii were our less tolerant populations. In general, these populations, Arizona and Hawaii, were very similar in their response to MG, as were Virginia and Iowa. So we included these populations as 2 groups. We essentially grouped Virginia and Iowa and Arizona and Hawaii together for this. And for our treatments, I gave control birds a sham inoculum that was just sterilized medium. And then experimental birds got a low virulence isolate. And we had 5 birds per group per population for this experiment. And I looked at gene expression in 2 different tissues, in the Harderian gland, which is a gland that animals with nictitating membranes have. And in birds in particular, this is really important for localized immune response. And then I also looked at gene expression in the spleen. And I looked at gene expression 3 days after we experimentally infected the birds with MG. And the reason we chose these particular tissues and this day of infection is because previous work done by Michal and also by Jim and others has shown that the inflammatory signaling and other signaling really does peak very early in infection. So it seems like early differences in gene expression might have a big impact on the eventual outcome of infections. So I want to start by looking at the differential expression of genes in Harderian glands. So these were the glands by the eyes that are important for localized immune response. And I was able to identify hundreds of differentially expressed genes when we compared our uninfected controls to our infected birds from each of our populations. So the overlap here, these 50 genes were differentially expressed when we compared uninfected to infected birds in all 4 of our populations. So regardless of how tolerant birds were, these genes showed up in all 4 of our populations. There were 39 genes that were only differentially expressed in our tolerant populations. So comparing control and MG infected individuals in those tolerant populations, there were 39 additional genes. And then an additional 371 genes were differentially expressed only in our intolerant populations. So there were many more genes that were differentially expressed in these intolerant populations compared to the tolerant populations. And these genes really do represent good potential candidate genes that may contribute to or control tolerance to MG and house finches. And we were able to annotate about 60% of these differentially expressed genes and found that they were really involved in myriad physiological processes. So the majority of our genes we were able to put in 4 rather large categories. So cellular processes, immune response and disease, metabolism, and also signaling. And here on our y-axis, I'm going to show you the number of pathways that are that have up or down regulated genes. So anything above 0 is a pathway that has upregulated genes. If it's below 0, that indicates that pathway has downregulated genes compared to uninfected controls. So you can see across our 4 major types of genes, we do see that there are many pathways in each of these, but really quickly we notice that less tolerant populations have more pathways with differentially expressed genes. This isn't surprising since they have more genes overall that are differentially expressed. But really the immune system genes were the majority of these, and that's what I want to focus on now, especially given the topic of this, this conference. So here I'm going to show you the number of immune system pathways that have differentially expressed genes in them for each of our 3 groups, our less tolerant, more tolerant, and then all tolerant group, or all populations groups. So in less tolerant populations, there were 9 immune pathways that had upregulated genes. This included things like tumor necrosis factor signaling, NOD-like receptor signaling, antigen processing and presentation, IL-17 signaling as well. And then one pathway was downregulated, and that was T-cell receptor signaling pathway. In our more tolerant populations, there was only one pathway that had an upregulated gene that was unique to more tolerant populations. And this was Th17 cell differentiation, which makes sense as this is associated with mucosal barrier protection. And then in all of our populations, we had a couple of pathways that had upregulated genes, including complement cascades. And then there was a natural killer cell mediated cytotoxicity pathway that had downregulated genes. And then there were pathways of cytokine receptor interactions that had both up and downregulated genes in all of our populations. So that was all for the Harderian gland. So that was more of a local response to infection. So next I want to take a look at the spleen, which will be at least comparatively more systematic response to infection. And again, I want to show you the number of genes that are differentially expressed between control and infected individuals in our 3 different groups. And one thing to notice right away is that there are more genes differentially expressed this time in our tolerant population compared to our intolerant. Again, we had genes in pathways across all kinds of physiological processes. But again, the majority were in immune system pathways, and that's what I will focus on. So for our intolerant populations in this case, there were only a couple of immune pathways that had upregulated genes, and this was TNF signaling and IgA production, and then a lysozyme pathway was downregulated. In our more tolerant populations, there were a few pathways with upregulated genes, including pathways that lead to phagocytosis. But really, what's interesting here is that there are more pathways with downregulated genes in the spleens of our more tolerant birds. So things like T cell receptor signaling, antigen processing and presentation pathways were downregulated in the spleen of infected birds in our more tolerant populations. And then in all populations, we had an equal number of pathways with up and downregulated genes. Upregulated gene pathways included things like NOD receptor signaling pathways, and downregulated included things like NF-kappa B. There were also 4 pathways with both up and downregulated genes, so things like chemokine signaling and cytokine receptor interaction signaling were included in these pathways here. So in general, that was a lot of information, but I want to kind of summarize this for you. In the Harderian gland, so that gland by the eye, that's a more localized response to infection, we see there are more genes differentially expressed in our less tolerant population compared to our more tolerant population that had fewer differentially expressed genes. And the less tolerant populations had more immune genes upregulated, including genes that contribute to inflammation compared to our more tolerant populations. We saw the opposite in the spleen, where there were fewer differentially expressed genes in our less tolerant populations and more differentially expressed genes in our more tolerant populations. And although there were fewer immune genes that were differentially expressed in our less tolerant populations, importantly, the more tolerant populations had more immune genes actually downregulated compared to upregulated. Compared to our less tolerant populations. So in summary, tolerance in this system at least does seem to be affected by genes in many different physiological pathways. So there might be very diverse mechanisms that are contributing to tolerance in this system. Differences in gene expression early in infection, so remember this is on day 3 of infection, might be really important in terms of determining the outcomes of infection. So how tolerant individuals might be determined very early. And then of course, given the differences between these two tissues in gene expression, it does seem important to consider both local and systemic immune responses. So doing more than one tissue when looking at gene expression differences between tolerant and intolerant populations could be very important. And finally, it seems that, at least in this system, dampened immune responses may contribute to tolerance. And in this case, our Western house finches, which were less tolerant, did seem to have a less targeted immune response to MG infection compared to the relatively more targeted response to infection that our Western, more tolerant populations had. So, of course, there's lots of questions remaining. This work always brings up new questions. So, for example, are there changes in these patterns throughout infection? Do we continue to see similar gene expression patterns as individuals progress through infection, or do they change? Are sequence differences affecting these? We don't know a lot about that. And then, of course, are similar patterns going to be found in other host pathogen systems? There's some work already that suggests maybe yes, maybe no. So it'll be really interesting to see how that works out in other systems, and if we see similar patterns in other host pathogen systems as what we found in the house finches and MG system. So I'd like to thank all of my co-authors, as well as everybody who helped collect all of these house finches and perform these captive experiments, and our funding sources. And I'm happy to take any questions I have time for at the moment, but please, if you'd like, you can write down my email address and send me questions that way as well. And thank you all very much for taking the time to listen today.
Wonderful, thank